Hi there, and welcome to Smith and Sheridan on Biotech, a podcast on the science and business of biotechnology, presented by me, Cormac Sheridan.
And me, Andy Smith. Hello, Cormac. How are you this week?
I'm good, Andy. How are you?
I'm well, thanks.
Are you ready to talk about PROTACs?
I'm willing to talk a lot about PROTACs, yes.
And that's it. There is an awful lot to talk about when you're talking about PROTACs. And I thought we'd call the session, or my kind of guiding little line into this was, where have all the PROTACs gone? Because a couple of years ago, they were ubiquitous in terms of the news.
That's a good word and a relevant word.
Yeah, and entirely unintended.
Oh, really? When I read that for the notes list, I thought ubiquitous, ubiquitin, ubiquitase. I must use that word more in my English. And well, there we go. Well, I must be more ubiquitous in my use of the word. And there we go. Straight onto my to-do list and straight off again in the space of a few seconds.
And purely serendipitous on my part. Yeah. But the PROTACs, they were really, really interesting. PROTACs and other approaches to targeted protein degradation and degradation also of things other than proteins. There's so much innovation in this space. And there's a ton of activity. I was doing news reporting about it all the week. It was like a little mini biotech cluster of all these interesting companies with interesting approaches and deals going down and the whole thing. And then suddenly nothing, nada, rien. So I thought it'd be interesting to kind of see what's been happening since. And what's been happening since, actually, is what needs to happen in biotech. The guys have taken their cash and they've come up with molecules and they've pushed quite a few of them into the clinic. And the lead program from Arvinas, and Pfizer is a partner in this, is in phase 3. 2 phase 3 studies ongoing in a certain form of breast cancer, which is estrogen receptor positive and HER2 receptor negative. And that's an agent which I'm going to struggle to name. It's vepdegestrant. I say that questioningly. I'm sure I might have mispronounced it. But this is an agent that has already shown activity. There's been some phase 1b data published, and it has activity in heavily pre-treated patients. But anyway, that's kind of where we are now. And I know Arvinas recently did a deal as well with Novartis, which is interesting too, 150 million upfront for a second generation oral PROTAC, which degrades the androgen receptor, which is a big driver of prostate cancer. But anyway, without kind of getting into the specifics just yet of all the various programs, I mean, we should obviously take a look back at how this field has developed. And
even we might have skipped over it already, but your thumbnail sketch of what actually is a PROTAC and why it's so exciting.
Yeah, because the ubiquitin proteasome system is a very complex, heavily regulated pathway within all cells for eliminating proteins that
within all cells, you say. I wonder why they called it ubiquitin then.
And when I say all cells, of course, I'm sure some person can come up with some counterexamples. Oh, it's not present in these.
No, I was thinking of one just for this, a bone cell or osteoclast or something.
Yeah. But anyway, in many cells. So it breaks down, unneeded, misfolded, or damaged proteins. And it does so in an exquisitely regulated fashion that you get this kind of enzymatic cascade. Here it is, folks, the ubiquitin protein, which is a 76 amino acid protein, highly conserved in the mammalian kingdom, I think, and ubiquitously expressed. And it's attached in a sequential fashion. You have a ubiquitin activating enzyme, transfers it to a ubiquitin conjugating enzyme, which then passes it on to a ubiquitin ligase, which then passes it on to a target protein. And when the target protein accumulates or develops a ubiquitin chain of several of these ubiquitin residues, it then enters the proteasome and where it gets broken down.
And that process is not just a one-way street, though, is it, Cormac?
No.
There's ubiquitinase activity and there's de-ubiquitinase activity.
Yes.
So if your protein
AKA
is looking...
Dubs, which is appropriate for people who will either live in Dublin or who are fond of a certain iteration of reggae in the mid-70s.
If there's a protein floating around in a cell that's looking a bit ragged, but not that ragged that it's still usable, then it will be de-ubiquitinase and then it will be functional again.
Exactly. And I think as well there's some really interesting research, which I've only barely scraped the surface of, in terms of understanding the ubiquitin code. And so I think there's even other biological functions as well. The whole thing is incredibly complex, cell biology is. And there are only 2 ubiquitin activating enzymes. There are 35 ubiquitin conjugating enzymes, and there are about 600 ubiquitin ligases. And they're divided into a bunch of different classes. And the clever thing that drug developers, and I think the key paper and the key developers who kind of kickstarted this field would have been Craig Crews and Raymond Deshaies and Kathleen Sakamoto. They reported the very first PROTAC more than 20 years ago, 2001, in PNAS, July 17, for those who want to get the reference. And they developed the first one in a kind of a rational fashion. But the field was dormant for a very long time because these are large, clunky molecules.
Yeah.
They have poor cellular permeability, poor solubility. They're much, much bigger than the kind of small molecules that are routinely used to inhibit protein function. But there was then another key finding, and this actually came from scientists in Celgene. They had these immunomodulatory drugs, which were very important, especially in
multiple myeloma.
And they had this extraordinarily interesting finding. They published some papers back more than a decade ago, or about a decade ago, that lenalidomide, which was Revlimid, exactly, multi-blockbuster drug, which was a derivative, and a better derivative, a safer derivative of thalidomide. They obviously had some sort of a phenotypic manner of picking up that drug. They didn't really actually understand the mechanism until after it was approved. But they found that it worked by binding the E3 ubiquitin ligase called cereblon.
Uh-huh.
And then they followed that study up with another study suggesting that those drugs work by ramping up the degradation of two immunosuppressive transcription factors, Ikaros and Aiolos. And that work was done in sort of about 2014 or so. And that really kickstarted the field proper. That's when money started flowing in because people realized that here was a way of getting a handle on protein targets that are not druggable by conventional means because they lack the kind of binding pockets or the chemical toeholds that ordinary small molecules needed. So these PROTACs, proteolysis-targeting chimeras, they basically have three basic components. One bit that latches onto the protein of interest, one bit or ligand that attaches to the E3 ubiquitin ligase, and a linker then that just joins these two. And the molecules are known as usually heterobifunctional molecules because they contain two active areas, and they're heterofunctional because they each have different functions and different binding properties. And there are other iterations which we can go into later on, but that's the basic idea.
The original first generation PROTACs, they were a bit stymied by, didn't they have a whole big chunk of E3 protein on the end, and that made them a bit less amenable to be drugs than the smaller molecule stuff that binds to E3 now and makes them more druggable. But you don't have to have these days drugs that bind to active or allosteric or other sites. All the PROTACs are doing is bringing the protein you want eliminated from the cell that perhaps causes disease in close proximity to the ubiquitase system that degrades it. And that was, I mean, we've known about this for a while. You know the Roche hemophilia drug, the first one actually to treat hemophilia that wasn't a blood clotting factor, but it just brought two clotting factors together in the same way as the PROTACs bring the ubiquitase protein and the target of interest together within shouting distance, if you like, by use of that linker.
Well, except though that, of course, you do have to harness the ubiquitin proteasome pathway.
Yeah.
You do have to get attachment of ubiquitin moieties to your protein of interest. But Craig Crews has resurfaced in a company called Halda Therapeutics, which I covered a couple of years ago when they raised some cash. And they don't use the proteasome system at all. They're developing things called RIPTACs, right? And they have what's called a hold and kill strategy. And the focus is on cancer, although you could potentially apply it elsewhere, maybe, but cancer is the obvious thing to go for. So RIPTACs are heterobifunctional molecules as well, but what they do is that they contain a ligand that binds an intracellular protein that is found in tumors, but it may not necessarily be the oncogenic driver, but it's just a marker for a tumor cell, but it's an internal marker, not an external one.
Yeah.
And the other part of the PROTAC, because it is also heterobifunctional, it comprises with a ligand that binds a protein that has an essential function within the cancer cell.
Yeah.
So again, it may not necessarily be anything to do with cancer. It could have something to do with just ordinary cellular metabolism.
Yeah.
But by setting it up in close proximity with the target protein, what you're doing is interfering with its function. So that what you get is rapid cell killing. Yeah. And they're still preclinical as far as I know. But what they're driving, essentially, they're kind of creating a novel or neo-protein interaction, and they're kind of locking this protein that's essential for cellular function into some sort of a state where it cannot discharge its function. And of course, the clever part is understanding how you set up those pairs in ways that are going to work. So there's a lot of complex structure-activity relationship going on, you know, but very nice idea. So back to the PROTACs, we're still in the early days. I mean, we've seen some, as I was saying, some data reported from the Arvinas Pfizer program, which is by far the most advanced. It's the only phase 3 that I could find. There may be others.
Yeah, no, me neither. But I mean, we are pushing the boundaries this week, aren't we?
We are.
I mean, we just had a quick discussion earlier on about close spatial proximity, which is a concept within drug discovery. And we're still talking about almost at later stage PROTACs, these 3 domain regions that allow you to remove a protein that's causing disease in your body. And I know we're going to get onto this in a second, but there are some downsides because you can't just do it on any floating protein. And there are issues associated with the size of the molecules and things. But we'll get on to that.
And the safety is going to be important, obviously, because these are kind of big weapons.
Yeah. And they're not blunt instruments either. I mean, they're supposedly not blunt instruments because the functional end that targets the protein you want removed from the body, that's quite specific, right? But then actually catalyzing the ubiquinase process is responsible for the linker that links the 2 parts. And if that's not permanent, or we have the same issue with drug antibody conjugates where we've gone through cleavable and non-cleavable and partially cleavable linkers. But if you cleave it in the wrong place, you're going to get protein degradation where you shouldn't.
Yeah. On the upside, potentially, is that these as well aren't eliminated the way that regular drugs are.
You see that as a positive, Cormac. I see it as I don't want this floating around in my system forever. I just want you to cure that one cancer.
Yes, then, true. And obviously, if it's not toxic, that obviously is an issue, but that you're getting this constant turnover of an unwanted protein, that could be interesting. But what strikes me too, though, is that it's early days, and I do want to just alert people's attention to a terrific review that's just published. It was published in just a month ago, the 2nd of April, 2024, written by 2 scientists at the University of Manitoba in Winnipeg, Canada, Kylie Rutherford and Kirk McManus. And it's called PROTAC's Current and Future Potential as Precision Medicine Strategy to Combat Cancer. Beautiful insights in there. One of which, a key insight, was that of the 600 or so ubiquitin ligases, only 13 so far have been harnessed in PROTAC designs. So I think we're still looking at a massively untapped resource. Now, again, you've said it before, very often the first one in is the one that works. So it remains to be seen, because obviously what people want is functional drugs that have oral bioavailability at relatively low concentrations, because it's believed that you could dose these at lower concentrations if you can get them into the cell. You don't need a massive dose within the cell, but you need to fire an awful lot of the system to get them into the cell in the first place. And that's where you get the off-target toxicity.
Well, you've got to get them to the right cell and then into the cell. So it's myriad.
Yeah, well, they're oral drugs, right? So they will have, say, in the case of cancer, these won't necessarily be external, they won't be necessarily binding receptors. But the rush, though, in the last decade or so has been huge. I mean, we've mentioned Arvinas and Pfizer. Chimera has done a big deal with Sanofi, and they're in phase 2 into autoimmune diseases, atopic dermatitis and hidradenitis suppurativa.
The actual foray into autoimmune looked interesting to me, but I thought it was quite ironic that, you know, some dysfunction of the ubiquinase system results in some cancers, and then most of the indications that it's been proposed for are a cancer. So in your face cancer from the ubiquinase.
Yeah, that's true. Because, I mean, 15 years ago, we're all talking about proteasome inhibitors, and 20 years ago. And there was a whole, Millennium Pharmaceuticals made their name on that stuff. And they were big guns, but they were pan-proteasome inhibitors. And that's why this has just a greater degree of finesse, because you're not shutting down the whole thing.
Which I can see appeals to you, Cormac.
Yeah. I like selectivity. So chimeras enough here and there, and their compound KT-474, it degrades IRAK4, which is some sort of an immune signaling molecule. And chimera also has a selective degrader of STAT3, a transcription factor that can act as a driver of certain cancers arising from activating mutations or oncogenic fusion proteins. Also, STAT3, newly into the clinic, chasing that one as well, is Devidium Therapeutics and Bayer. Bayer bought them for whatever it was, 1.5 billion and 500 million milestones back in 21. So they've just gotten in as well with DVD 130850, and that's in phase 1/2 in solid and hematological cancers. And there is, as is often the case, a sort of a convergence on a number of key kind of targets.
Yeah. Prostate cancer.
Yeah. And molecular targets in terms of what to go after.
Oh, I see. KRAS and
KRAS, and there's GSPT1. BMS is going after that one. Biotech is going after that one. And also
HER2.
There's another company going after the GSPT1. It's Monte Rosa Therapeutics. They're into a molecular glue degrader rather than a PROTAC. And again, it's that sort of proximity type of approach where you create a sort of a composite surface that allows for this new protein-protein interaction to rise, and that's how you get that glue-driven degradation, which may be safer. Who knows?
Well, we are pushing the boundaries this week, aren't we? Because
we are.
Protein-protein interactions is a much bigger, wider field that, again, people have been talking about for years, but PROTACs is a good entry point for that.
Yeah, and they've gotten... Modules have put out some data, interim analysis of the dose escalation phase of their phase 1/2 trial. Their agent, MRT-2359, clinical activity across all 6 dose levels examined. And of the 6 biomarker positive patients, I presume the biomarker was something to do with GSPT1. They're going after MYC-driven cancers in particular. And they had 1 confirmed partial response, 1 unconfirmed partial response, and 1 had durable stable disease. And all the rest of it. So it's promising, but it's still early days.
Yeah, and it did relieve me a little bit, because the most advanced, the Avenus Pfizer product, in phase 3, but it's with another agent. So it's been tested in combination. And whenever you have a drug in cancer that you see clinical trials for, it's not a single agent activity. It starts to be worried that perhaps it's not good enough. But there is plenty of other data around that say that PROTACs have single agent activity. So I was less worried about that.
Yeah. And I mean, to be fair, obviously, you'd expect it. The phase 3 program that they have, I mean, it is rigorous. There's 2 studies, and one's comparing vepdegestrant with palbociclib or Ibrance, a cyclin-dependent kinase inhibitor, and with, again, the PROTAC. That's one arm, and the other arm is vepdegestrant and letrozole, the aromatase inhibitor. And then the second study is doing a head-to-head of vepdegestrant versus fulvestrant, an estrogen receptor antagonist that's been around for a while. So they're giving it a fair old testing there now.
And as we said from the very beginning, these are very innovative molecules with lots of complications and risks associated with them. So you want to offer patients something that's not just that very innovative agent, but so you're going to give them a combination with a proven drug in that indication as well.
Of course, and you're looking for obviously additional activity on top, and hopefully that's what
which by this time should be pretty well known. So anything on top of that should be obvious. And again, these patients aren't going to be the first line breast cancer patients.
No. And another outfit to report some data is Bristol Myers Squibb, aka Celgene. Obviously, they got into this through Celgene. They have an agent, CC9476, one of the many agents that's degrading the androgen receptor, which is kind of an obvious thing to do in prostate cancer. And they reported phase 1 initial results. They had prostate-specific antigen responses and radiographic tumor shrinkage at all dose levels, which is kind of a, table stakes, but it's positive. And
which is a bit of a last gasp validation for the old Celgene's approach to business development, where they did a scattergun approach of doing collaboration with almost any biotechnology company you can think of. And when Bristol Myers bought them, they shut most of them down. But this one carries on. You fire enough arrows, you'll hit something.
Absolutely. These are, again, heavily pretreated patients who progressed on a number of different agents, and they do have responses. 9 out of 20 patients, 25% remain free of radiographic progression with treatment ongoing at 6 months. That's just taken from a press release, I guess. So there's kind of There is activity. And I think the next 12 months is going to be very important. Next 12 to 18 months, that's when we're going to see more data coming through. C4 Therapeutics is another one of the early players, and they have a whole bunch of different agents in the clinic. They have one this is actually an interesting one to me. It's called CFT7455 because it's actually targeting the two transcription factors, Icarus and Ailos. I presume they're Greek islands. And this is a kind of almost to me a reiteration of Revlimid or lenalidomide, but presumably with better characteristics and more activity. So I think that's very interesting. And that they also have something going after BRAF V600 mutated solid tumors they're going after. Yeah, that's CFT 1946. Collagen is another crowd in there, and they're going for an oral pan-TRK degrader in solid tumors. So there's not all, but a lot of it, as you can see, is cancer, and for good reason in a sense, because problematic proteins are a particular hallmark of cancer.
Yeah.
But what fascinates me,
even aberrant proteins, which might make the selectivity index even better, because if they're not normal, then just get rid of them. But I feel the need to not so much pour them cold water on it, but just a calming influence on your excitement, Cormac, because
my partially rational exuberance, maybe.
Because these are exciting technologies that you can remove a protein of disease-causing ability in the body, sure, but not every protein, right? It's only those in cells, and you've got to get the PROTAC in cells, and PROTACs are reasonably big molecules, and they're polar, and you talked about oral bioavailability. So yeah, it's not the panacea for everything, but it's nonetheless exciting.
If you don't want a PROTAC, I can offer you other alternatives. I can offer you an ATAC or an ATAC. Yeah. This is a really interesting technology developed by Avalar Therapeutics. And they're doing targeted degradation of extracellular proteins.
Oh, even? Excellent. Okay.
And I think this is really cool. They've got basically the ligand is, instead of binding an E3 ubiquitin ligase, these are extracellular drugs, obviously, and they bind an asialoglycoprotein receptor expressed in hepatocytes. Good. So that they have a beta ligand that picks up on the circulating protein that they want to get rid of. And then that attaches itself to the asialoglycoprotein receptor. And the whole thing gets taken up into the hepatocyte by endocytosis. And hey presto, protein degradation follows in the endolysosome.
Do you know, I've just had an idea for a science fiction film where the criminal was let off because he designed an ATAC that changed his blood protein components away from what they were before. So any evidence at the crime scene was not him. So he got let off.
I think he might have been dead, though. I mean, I've been able to enjoy his freedom.
Yeah, you destroy your HLA system and you probably got a lot of,
I think that's more than a big gun. That's actually sounds to me like molecular obliteration. Well, anyway, we can work that one out in another setting. But they're talking about using this for autoimmune and neurological conditions. And then there's another really interesting one, Lyceal Therapeutics. They're developing LITACs, lysosomal targeting chimeras, which is a similar sounding idea. So they're using an antibody or an antibody fragment, and that's directed to the target, and that's conjugated to a glycopeptide agonist that activates a cation-independent mannose-6-phosphate receptor on the lysosomes. So that's, again, a protein internalization and degradation approach.
Have you ever woken up from a dream and think, that was real? And I think when I first heard about PROTACs, someone told me about it, or I actually assumed the small molecule for PROTACs that actually binds to the target protein was an antibody. I thought that's how you do it, but obviously not.
Yeah, and I presume it's to keep the size down, but it works though if you're dealing with an external or cell membrane type of settings where you want internalization. And they've explicitly said like they're going after difficult to target membrane proteins, circulating immune complexes, autoantibodies or neutralizing antibodies. And one of the founders there is Carolyn Bertozzi, a recent Nobel laureate. So heavy-hitting kind of people involved there. So I just think it's really interesting that, and actually another one is PAC Therapeutics, although they've been quiet of late, but they set out, they raised some cash some years ago. They were harnessing the autophagy pathway, and they were going to get rid not only of proteins, but of, say, damaged organelles and a whole range of molecular substrates that don't get swallowed up by the proteasome pathway. So it's flying the area, and it's at the stage now, obviously, where you need to deliver data, and the ones that work, the technologies that work, the approaches that work will presumably generate data, which will generate cash and generate deals. The field will progress. And these are difficult drugs, you would assume in time that they could maybe simplify the structures, but there's only so much you can do with chemistry, right? But when you have a winner, that's what generates the cash that enables you to do further refinement.
And you can't have that winner unless lots of companies try and experiment with it.
Yeah. And like there's been a ton of deals done here. Big Pharma has really piled into this space.
Yeah.
So you've had deals with Pfizer. Arvinas have done deals with Pfizer, Genentech, Roche. C4 has done work with Biogen and Roche. Bayer has gone with Arvinas. They've actually done a joint venture for agriculture, Earth Bio. Earth spelled OERTH. Earth Bio. Chimera has done some work with Vertex. Nurix, another important player here. They've done some work with Gilead. So I haven't checked where all of these are at as yet. And Eli Lilly is in there too. They've done work with Seed, and AbbVie's done work with Frontier Medicines, another important company. There's a nice summary, actually, Biopharma Dealmakers from Nature. This is May 2022, just to give them their credit.
So it is an exciting area, and it is so exciting with so much potential. And again, with so many people working it, that we've not talked about it as a bandwagon yet, probably because it's still unproven. But I
tell you why I don't talk as a bandwagon. It's so damn complex.
Yeah.
To be in any way a credible player here, you really need to know what you're doing. And I respect the very, very clever way that you can go from PROTAC to these other related modalities. I love the way that people are harnessing different aspects of cell biology to try to achieve a therapeutic outcome. And we'll learn. There's a long road to go in terms of the clinical translation of all of this stuff, but it's underway.
Yeah, and can I leave you with something I thought of, an everyman definition for PROTACs? Spring cleaning for aberrant or disease-causing proteins. How's that?
I think that's on the money. I'm going to leave it with that. Say it again. Spring cleaning for
aberrant or disease-causing proteins.
On that note, bye till next time, Andy.
Cheers, Cormac. Bye-bye, everyone.